Hello friends, welcome to my channel Same's Biology. Today in this video lecture we are going to understand the packaging of DNA. In my previous video lecture we have studied about the structure of DNA and particularly the Watson and Crick model of DNA. So in this video let's understand about the packaging of DNA. So how the DNA is packaged inside a very teeny cell, and we know the size of the nucleus is again very small as compared to the cell, and inside that small nucleus how the long thread-like structure DNA is arranged. So we will study about the packaging of DNA. So the human genome has 3 billion base pairs packed in 23 chromosomes. So we know the genome means the total number of genes in n number of chromosomes. So in haploid cell in n number of chromosomes there are 23 chromosomes, means inside 23 chromosomes there are 3 billion base pairs. We are talking about human beings. So in diploid cell we have twice the number of 3 billion, that is 6 billion base pairs are present. Okay, so each cell, by calculating from this, each cell will have 2.2 meters of DNA. So the length of DNA in a single cell is 2.2 meter. Clear? So 2.2 meter of DNA is present, it's arranged in a very teeny nucleus. So we need to know how so long DNA, such a long structure is arranged inside a teeny nucleus. Human has 50 trillion cells. So it is an approximate value. So the human beings have 50 trillion cells, and each cell has around 2 meters of DNA. It will make 100 trillion, approximately 100 trillion meters of DNA are present in human beings. In a single human, 100 trillion meters of DNA are there. So if we will calculate the distance between Sun and Earth, it is about 150 billion meters. Clear? So we need to understand what is the length of the DNA present in our body. That is why I made a comparison. So Sun to Earth, the distance is 150 billion meters. So if we have 100 trillion meters of DNA, that means if we will make it, we will know the distance between Earth and Sun. If we will make the DNA arrange the DNA from Earth to Sun and back, then the DNA in a single human will be so much long that it can cover the distance of Earth and Sun and back, from Earth to Sun and back 300 times. Clear? So from Earth to Sun the DNA covers and back, it can go to Sun and come back 300 times. So much long DNA is present in our body. So how they are arranged in our cell? What is the way how such a long structure of DNA are present in our body? So what is the way the DNA is packed inside the nucleus? Yes. So to understand the packaging of DNA, let's know what is the structure by which the DNA is arranged so compactly. So it is nothing but the histone. So the histone protein plays important role in this. So what is histone? Histone is made up of 4 different types of proteins. They are H2A, H2B, H3 and H4. So these are the 4 different proteins, 4 different parts of the histone. So remember, these 4 parts, these 4 proteins are present in 2 number. So in pairs they are present. So histone protein is a structure like this. We can draw it like this. There are 4 different proteins and each protein have 2 components. So they are present in pairs. This one is H2A, this one H2B. So another H2A is present at the back side, H2A. Another H2B is present at the back side H2B. This one H3, H4. Again, they also have their components H3 and H4. So they are present in pairs and makes the structure of histone protein. That is why this structure is called the histone octamer, because 8 different subunits are there to make this histone protein. What are the 8 different subunits? H2A, 2H2A, 2H2B, 2H3 and 2H4. Okay, clear? Now another protein is also there which is associated with this octamer protein. That one is H1. So H1 is there which is attached to this octamer structure. Clear? But it will not take part in the process of hereditary, in the process by which the character from parents are transferred to the offspring. Hereditary transmission. In hereditary transmission, this H1 will not play any role. Clear? So how DNA is arranged with the help of the histone protein? Let's see. So we have the histones, which are, the histones are positively charged. The histone proteins are positively charged due to the presence of 2 different amino acids, the lysine and arginine. Due to presence of lysine and arginine, due to the presence of these two amino acids, lysine and arginine, the histone protein becomes positively charged. Okay, so the histone protein is positively charged, and we know the DNA is negatively charged. The DNA is negatively charged. Why? Due to presence of phosphate. Due to presence of phosphate in the sugar phosphate backbone that we have studied in the structure of DNA. In the sugar phosphate backbone of DNA, the phosphate is there. So due to the presence of phosphate, the DNA is negatively charged, and due to the presence of these 2 amino acids in the formation of the histone protein, the histone is positively charged. So we know that always positive charge attracts negative charge. That is the reason why the histone binds with the DNA, it attracts the DNA, and a complex structure is formed inside the nucleus. Clear? So now let's see how this histone protein is attached with the DNA. So we have the DNA. DNA is a thread-like structure. The structure is attached with the histone. DNA is attached with the histone to make chromatin. The structure of chromatin. So chromatin is made up of many nucleosomes. So DNA and histone together forms nucleosome, and these nucleosomes together form the structure of chromatin. Nucleosomes are the units of the chromatins. Clear? So we have already know that there are 9 proteins. 9 proteins in histone. These 4, H2A, H2B, H3 and H4 are in pairs, and H1 is single. It is present in single, only one H1 protein is present. So how this DNA is bound to the histone? Let's see, and forms the nucleosome. Okay, so the structure nucleosome is formed like this. This is the histone aptamer. We have it here. It has 9 proteins. The aptamer has 4 into 2 means 8 proteins and 1 H1. So this H1 will not come under the aptamer structure. So this is the aptamer, and if this is the DNA thread, then it will go like this. It will coil around the aptamer protein, the histone protein, and will come, and this is the DNA, and this will come and bound to, it will bound to another. Look here, if I will do another structure of nucleosome here, the DNA will come and bound to another histone and will go. So the DNA thread coils around the histone protein 1.75 tons. 1.75 tons means, look, it came in contact with the histone here, one complete circle, then 3/4, and then next to another nucleosome. Here also it will complete one circle, then 3/4, and will go to another, and like that. So the structure will be like this. So the structure, if I will make here, it will be like this. It is the thread of DNA. This is one nucleosome where it complete one circles, next circle 3/4, then another 3/4, then it will come to another nucleosome, complete 1 3/4, complete 1 3/4, like this. It will go to another nucleosome, complete 1 3/4, and this process will go on. So this is one nucleosome. So around 200 nucleosomes are present in a single chromatin, and the thread, the DNA that links the two nearby structure nucleosomes, are known as linker DNA. So the thread of DNA here, it is the linker DNA, because these pieces of DNA joins one nucleosome with the nearby nucleosome. So this is called the linker DNA, which is around 20 base pair long. It is 20 base pair long, and the part of the DNA which is present surrounding the aptamer has 146 base pair. So the length of the DNA surrounding the nucleosome, surrounding the histone, is 146 base pair, and the piece of DNA that connects two nucleosomes are the linker DNA, which is only 20 base pair. So total of 166 base pairs are present in the single nucleosome and the connector. Okay, the connecting piece of DNA. So this is the typical structure of a nucleosome, and it makes the combination of around 200 nucleosomes makes the structure of chromatin. Clear? So the nucleosome has histone protein and the DNA. So they together forms a nucleosome, and many nucleosomes are present in a chromatin. So if you will study the structure of chromatin, it will look like this. This is a thread, and if you will darken some places of the thread, the beaded like structure in the thread are known as nucleosome. So these are the beaded like structure which are called the nucleosome, and the intermediate, the place between the beaded-like structure are the linker DNA. As I have told you, it connects the nearby two, the consecutive nucleosomes. Clear? So another thing we need to know, sometimes non-histone proteins are also required. Non-histone proteins or non-histone components. Why? The non-histone proteins or non-histone chromosomes which are made up of non-histone proteins, they are needed for more complex chromosome. So for the formation of more complex chromosomes, packaging of higher level requires additional set of proteins which are non-histones. Okay. Another term we need to know, one more thing we need to understand here. In nucleus, some region of chromatin are loosely packed. Some regions of the chromatin are loosely packed or not so much thick. They look light and they are known as euchromatin. So the loosely packed area of the chromatin are called euchromatin. Remember, this is very important term. These euchromatins play important role during transcription. So they are transcriptionally active. Okay. So loosely packed euchromatins are transcriptionally active and the densely packed heterochromatins are inactive. So let me write here the densely packed heterochromatins. The densely packed chromatins are called heterochromatins, and these heterochromatins are transcriptionally inactive. They do not take part in the process of transcription. So they are transcriptionally inactive. So heterochromatins are the densely packed region of the chromosome or the chromatin, and euchromatins are loosely packed and they take part in the process of transcription. One important thing we need to know here about the structure of nucleosome or the octameric structure. This octamer structure where the DNA is bound to the 8 proteins. Here the H1 protein is also present, which helps in binding the DNA with the octamer protein. Roger Kornberg is the scientist who discovered this octameric structure of the nucleosome. So Roger Kornberg is known for discovering the structure, giving the structure of the nucleosome where 8 proteins forms the core protein or the octamer of histone, where the DNA is bound to it, forming the structure of nucleosome. So this is how the DNA is 2.2 meter long DNA in a single cell is packed inside the nucleus with the help of the positively charged histone, and it attracts the negatively charged DNA so that a nucleosome is formed, and they together make a compact structure of chromatin which makes it easy to place such a long DNA in a very minute nucleus. So this is all about the structure and the packaging of DNA and nucleosome. Thank you very much. Please subscribe to my channel STEM's Biology. Thank you.